Okay. As I said, I wanted to go back to this topic before we move. It's a short presentation, so it will not take much time, but will help us also to move to the next practice. So, as you can imagine, the core of RNA-seq analysis is the differential expression. So I'm going to again remind you about general concepts, the statistical methods, and tools. And the general concept is simple. No, a differential expression analysis is a statistical analysis in which you compare two groups and you test the expression. You test an hypothesis in which you check if two of one gene specifically is differential expression between two groups. Usually you have associated a p-value or a corrected p-value that can be a q-value, or in another programs will be the false discovery rate. And when it's below some certain threshold, usually 0.05 or 0.01, is considered that they have differential expression. No? So a good example is you have this quantification matrix. And quantification matrix, remember from the last class, can be anything. Can be counts, can be PKMs, can be TMMs. Depending on the methodology, can be anything. And then in which you have as columns the conditions and as a rows the genes, and then you will test row by row the hypothesis to see if they are differentially expressed or not. From the statistical point of view, as I mentioned last day, this analysis has a very low number of replicates. No, it's usually don't use 20 replicates. No one uses 20 replicates. So indeed, when you perform this statistical analysis, they have a very low statistical power. Also, I mentioned that you have different options to test differential expression. You have mostly, the models can be divided in two parts or two types. One of them are the linear models, and the other one are the cone-based models. No, I will talk a little bit about that. No, so you have the scaling factors, that is the normalization, and then you may not have gene filtering, and it's when you're starting to apply these models until you arrive to your statistical test. You will have this point null to test the hypothesis of each of the genes, and you test again a threshold. You may correct the p-value later on, and I will explain a little bit about that, or you can do some modifications. But mostly, this is the methods. So what are these methods? Well, most of the statistical methods are parametric methods, because you have a small size, sample size, as I mentioned before. You only have between 3 and 5 replicates. So you usually apply a parametric method. Of course, you have other methods that are not parametric. Just as a reminder, parametric statistics is the branch of statistics in which assumes that sample data come from a population that can be adequately modeled to a probability distribution. Okay, so parametric methods, mostly the thing that you do is you test if or compare with the expected distribution that you should have. So your gene expression fits in this expected distribution is not differentially expressed, but it doesn't fit is when you can assume that you have a differential expression. As I mentioned in the last section, you have two different types of models. You have linear models or cone-based models. For linear models, first, of course, you transform the data in log data. And then, well, initially, these kind of models were suboptimal. Okay, the simple models that you can apply. But then these models has been improved. So you have the general linear models or with estimation of the dispersion parameters like MM. I don't want to go into much detail, but just let tell you that not any simple linear model can be applied to test the differential expression in some RNA-seq experiment. No, so you need to this model need to be in some ways robust. No, so the model that usually has been implemented for that is the GLM or the general linear model. The other problem is that because the dispersion, and when I'm talking about dispersion, I'm talking about in some ways how the data disperse no, in these populations, if they are clustered or they are more dispersed across the axis of the different variables that you have. The dispersion not usually need to be estimated to improve this kind of models. The second one are the cone-based models that are the most common ones. Okay. Yeah, usually the variation between technical replicates follow the Poisson distribution, but usually we don't use technical replicates in RNA-seq. It's more common the biological replicates. So, and the biological replicates usually follows this gamma Poisson or the negative binomial distribution. No? So more or less, as I said, you compare with these distributions in order to check if your gene is differentially expressed. As I said, I'm not going to go into the details of that. Just I want to give you that kind of a brief overview. So once you test this analysis and then you have this p-value, the next thing is that usually these kind of methodologies are enriched in false positives. Okay, it's quite normal to have a lot of false positives. So it's quite common also to run this false discovery rate of Benjamin-Horvech correction in order to minimize the false discovery. You will see that when you are running this test, you usually have two values. You have the p-value and you have the q-value or the FDR. The p-value, when you are using p-value, you will have a much higher number of differentially expressed genes. But as soon as you go to the FDR, sorry, or the q-value, you will see that this number will drop off dramatically because you are, in some ways, removing false positives. Of course, these things will depend of how much variant do you have in your sample. So the problem is that sometimes, if you apply the false discovery rates as you should, you may not have differential expressed genes. No? You will see that too. Well, that is an important thing. So sometimes, I mean, to apply the false discovery rate is highly recommended, but also depends about the variation that you have between your replicates. If the variation between your replicates is too high, and because sometimes it's difficult to minimize this kind of variance, then maybe false discovery rate is going to kill, okay, the experiment, or at least this differential expression analysis. Being said that, also, initially I said that most of the tests that you apply are parametric, but also you can use alternative model like Bayesian interference or non-parametric test. So you have also that option. Sometimes they have some of them can be applied. And then it's also common not to use only the dispersion to estimate if you have differential expression, but also you can use the dispersion to estimate the log fold change. So what is the log fold change? Is usually when you are running an analysis, you are comparing two conditions. So at the end, you want to see if you have an overexpression or an underexpression. You compare the expression of the sample 1 with the expression of the sample 2. So you divide one by other. So for example, you have here in the sample 1, you have by average, let's say 10 FPKMs, and in this expression sample 2, you have 5. 10 divided by 5, you will have 2. Okay, so that is the ratio of the fold change. We usually don't work with fold change as it is. We work with the logarithmic scale. And the reason is because when the numerator is bigger than the denominator, of course, you have values that are more than 1. But when the numerator is smaller than the denominator, you will have value from 1 to 0. So in some ways, this scale will not be symmetrical. Okay, you have from 0 to 1 when it's underexpressed, and then from 1 to wherever when it's overexpressed. So in order to have this thing symmetrical, we do the logarithmic 2 of this ratio. So below 0 will be underexpressed, and up to, over 0 will be overexpressed. Okay, so that is a common. I didn't have a slide about that. That's my error, but that is an important concept too, no? That we work with the logarithmic fold change. We will see that in the exercise. And then, of course, you have more refined methods in which you can account for unobserved effects. So you have some correction. I put here two examples, two methodologies. But this is surrogate variable analysis or the removal of unwanted variation. Okay, but as I said, that is up to the goal of our course. So in this table, I explain all the, well, I put the example of the different models, okay, as well as some of these programs. I have a more reduced table, but as I said, it's, in some ways also I wanted to put this table from this publication because it told you in these notes on the testing strategy what is the kind of test that you know. So just to have you for your records. I will still talk in tools what are the most common ones. Other thing that is also worthy to mention is that sometimes we don't test differential expression or differential gene expression, that is DGE, but we just test the differential transcript expression or the differential transcript usage. The difference is that the differential transcript expression, you look for transcripts. Meanwhile, the differential transcript usage, you quantify exons and then you average the exons, okay, on the potential transcripts. Okay, the first one just you work with transcripts, but the second one you work with exons and then you modelize the transcripts. Tools. So that is the practical part. So if you want to do this kind of analysis, what tools you want to use or you can use? Well, this is the most common tools. Okay, this is not an extensive list. This is not an extensive list either, but at least this is a list of tools that are the most used one. So if you want to use this Poisson distribution, the usual tool that you use is DefSec. That is, I think that is an R package, but has the limitation that ignore the biological variation. So in most of the cases, you don't want to use it. Then very popular ones are HR and DSEC2 that have this negative binomial model. Remember parametric binomials, and then they have the limitation that of course you only can compare pairwise, but as I said, they are the most famous ones, especially DSEC2. Well, that is the most popular one and give you good results. If you want to do a Bayesian approach, and Bayesian approaches sometimes are good, you use BiSec, but still it's a computation intensive. We are not going to see that. And then you have this NoiseSec that is a non-parametric analysis that sometimes is worthy to use it. So for log counts, it's good, worthy to use it too. Then you have also two that are very popular, especially because they derive from this very popular pipeline that are Kiplinks and Balgo. Kiplinks use this Poisson distribution and work with FPKMs, and then Balgo use linear models and also work with FPKMs. So that is the more common one. What is the thing that you should use? Well, it depends. No? Some people, for example, and this is also an review because they test different tools with different differential gene expression analysis. No? So they use for mapping tools like BWA, Top Hat on Bowtie, or then these pseudo assemblers, pseudo mappers that are Callisto and Salmon. I'm not sure where the is here. And then they do some quantification with HDSec or maybe with HR or NoiseSec, depending. They also have a way to quantify that. And then they're starting to run all the differential expression analysis with all these different packages and different conditions. Then they evaluate through positive to negative, false positive, and false negatives, and see which one was has the better statistical power. So to check what is statistical power, you can check different parameters. So this is through positive rates, specificity, positive predict value, accuracy. So when you put all these things together, you have this F1 measure that in some ways will give you the rank. Running that looks like the best method is DSEC2, followed by NoiseSec and Limaboom. Okay. I think that here they don't test Kiplinks and Balgo, but also let me tell you that usually in other publications they don't have good values. They are easy to use, but they don't have good values. So basically in these publications, looks like the method that is more recommended in this case is dissect2. Okay, and that we will see something about that too. Okay, so that, as I said, was a short summary of last day. Do you have questions? Because of course, when I asked you on Thursday, almost past 6, if you have questions, no one had questions. So do you have any questions about this presentation or any of the things that I presented here?
Actually, when you say pairwise comparisons, so it means that if I have to analyze 2 different sets of samples that are not different condition of the same sample, I cannot use dissect2. This is the thing, or I am misunderstanding something?
Well, it means that if you have 3 samples, you need to compare them 2 by 2. So let's put an example. Imagine that you have, well, in my case, no, just looking for one experiment that I was analyzing a couple of months ago. I have a plant and species, avocados, and I have 2 different accessions. I have one accession that is susceptible to a pathogen and another accession that is not susceptible to a pathogen. And then, of course, I have the controls, and I have these plants treated with the pathogen. So indeed, I have 4 possible comparisons. No, I can compare the 2 controls. I can compare the 2 infected. Or I can compare the control to infected in one of the accessions or the control to infected in the other accession. But I cannot compare everything at the same time. So that is one important limitation. One option is I can cluster them by 2. So I can say, I'm going to compare accession by accession. So I group control and infected, and then I compare them. So that is one possible pairwise comparison. Or I can compare infected with control and grouping all the accessions. That would be another comparison. Or I can break everything and compare, as I mentioned before, 1 by 1. So, but the problem is that I cannot compare everything at the same time. So that means pairwise comparison.
Got it. Thanks.
Yeah. And let me tell you, that can difficult a lot the experimental design. So you know, at some point I have this conversation with the statisticians that there are kind of a bit disalignment between what is the thing that you want as a user or as a biologist and what is the right way to run the statistical test. No, so I remember that I told them, no, but we need to compare everything. I said, yeah, sure, but you cannot do it. I said, no, but I want to compare everything. I need to compare everything. They said, no, with this kind of methodologies, you only can compare 2 by 2. Then you can assign some covariance in between other samples, but indeed these methodologies, the design that you need to do is compare 2 by 2. So that is a problem. Any other question? Okay, so you don't have any other question. I'm going to stop the recording.